Have you ever stumbled upon a piece of information that just makes your brain do a little happy dance?
I know exactly what you mean.
Prepare for some serious brain dancing today because we're going to be looking at some
Groundbreaking.
groundbreaking research.
Absolutely. We're going to delve into an experiment where scientists actually inserted a human language gene into mice.
A human language gene into mice.
It sounds like science fiction, but the findings offer some incredibly real and fascinating insights into our own biology.
It really does sound like science fiction. I mean, I'm looking at these papers right now.
Yeah.
And it lays out exactly how they did it, what the scientists observed, and where this kind of work could potentially lead us.
That's right.
So our mission today is simple: to understand this experiment, like the nitty-gritty, the surprising outcomes, and why it should pique your interest.
Absolutely.
All without needing a genetics textbook open in front of you.
Exactly.
Okay, so this language gene, FOXP2.
FOXP2.
What's the big deal? Why is this particular gene so important?
So interestingly, FOXP2, while often called the language gene, isn't a single on-off switch for language.
Right.
Instead, it's a critical player in the development of normal speech and language abilities in humans.
Okay.
We know this because when humans have mutations in FOXP2, it's often linked to significant speech and language disorders.
Okay, so if it's not the only gene involved, what makes it stand out? What makes it so special that scientists would target it in this kind of experiment?
Well, here's a mind-blowing fact. The human version of the FOXP2 protein differs from that found in our closest primate relatives, like chimpanzees, by just 2 tiny building blocks.
What?
2 amino acids.
2 amino acids.
And these seemingly minuscule genetic tweaks are believed to have played a substantial role in the evolution of our unique capacity for complex spoken language.
So you're telling me that a fundamental difference in how we communicate might hinge on just 2 tiny changes at the molecular level?
That's right. Think about that.
That's crazy.
Just 2 amino acids.
It's almost unbelievable.
It is.
That such a small difference could contribute to something as intricate as human language.
Right.
So the scientists pinpointed this key gene.
Yeah.
How did they actually manage to put a human version into mice? That sounds like some serious high-tech work.
It truly is a testament to modern biological engineering.
Wow.
The first crucial step was to create a humanized version of the FOXP2 gene.
Okay.
Now, this wasn't simply copying the human gene. They also included specific regulatory sequences. Think of these as instructions that tell the gene where and when to be active in the mouse brain, ensuring it's expressed in the right places at the right developmental stages.
Right. It's not just about having the gene present. It needs to be switched on and doing its job in the correct cells.
Precisely. Then came the actual insertion of this humanized gene into the mouse embryos. One method they used is called microinjection.
Microinjection.
Imagine using an incredibly fine needle to directly inject the human FOXP2 gene into the nucleus of a fertilized mouse egg before the genetic material from the mother and father fully combine.
Okay.
This allows the new gene to potentially become a permanent part of the mouse's genetic code.
Wow. That sounds like it requires incredible precision.
It does. And another even more precise technique they employed is CRISPR-Cas9.
CRISPR, yeah.
This technology acts like a molecular pair of scissors, allowing scientists to make very specific edits to the mouse's existing DNA.
Okay.
In this case, they could precisely alter the mouse's own FOXP2 gene to match the human sequence at those critical 2 amino acid differences.
Wow.
It's like performing a very targeted genetic update.
So 2 different but incredibly sophisticated ways of getting the human gene sequence into the mouse.
Yeah.
What happens after that? Do they just wait and see if the mice start chatting?
Laughing. Not quite. Once these genetically modified embryos develop and are born, a crucial step is to verify that the humanized FOXP2 gene is actually active in the mice.
Right. How do they check for that? It's not like they can give the mice a language test, right?
No language tests for mice just yet.
No.
They use molecular techniques. One is called RT-PCR.
RT-PCR.
Think of RT-PCR as a way of making a temporary blueprint of the active gene, checking if the gene's instructions are being read.
Okay.
And Western blotting is like checking if the cell is actually producing the protein based on that blueprint.
Gotcha.
Importantly, they also analyze brain tissue to pinpoint exactly where in the brain the human FOXP2 gene is switched on.
Okay, so they've confirmed that these mice have the human language gene active in their brains. What do they actually see in these mice? Let's start with the physical structure of their brains. Are there any notable differences in how their brains develop?
What's really interesting here is that they did observe some significant changes.
Okay.
The researchers found enhanced neuroplasticity, especially in the basal ganglia and the cortex.
The basal ganglia and the cortex.
Now, these aren't just random areas. The basal ganglia are crucial for coordinating movements like those needed for speaking, and the cortex is the brain's center for higher-level processing and learning, both essential for language development.
Okay.
So enhanced plasticity means these brain regions were potentially more adaptable and capable of forming new connections, like a brain that's more easily molded by experience.
So their brains were potentially more flexible in those specific areas, crucial for motor skills and learning.
Exactly. They also observed the formation of more intricate neural connections, specifically in brain regions known to be involved in motor learning.
Okay.
It's as if the wiring in those areas became more complex, potentially allowing for more sophisticated information processing.
Fascinating. Any other changes within the brain itself?
Yes, they noted alterations in dopamine signaling.
Okay, dopamine.
Dopamine is a chemical messenger in the brain that plays a vital role in both speech and the coordination of movements. The changes in dopamine levels in these mice suggested that the human FOXP2 gene was indeed influencing fundamental brain functions related to how humans develop these abilities.
Right.
Furthermore, they found a significant increase in the size of the striatum,
The striatum.
a part of the basal ganglia that, as we mentioned, is involved in movement and learning. This increase could indicate an enhanced capacity for complex motor coordination.
So changes in brain adaptability, more complex neural pathways altered dopamine levels,
Yeah.
and even an increase in the size of a key brain region. That's a pretty substantial list of neurological changes.
Absolutely.
But what about how these mice actually behaved? Did they suddenly become better at problem solving or display any new abilities?
Yeah, quite.
While they didn't suddenly start solving complex equations.
Okay.
The researchers did observe some intriguing behavioral differences.
Remember that mice primarily communicate using ultrasonic vocalizations, high-pitched sounds we can't typically hear.
Oh, yeah.
What they found was that the mice with the humanized FOXP2 produced different patterns of these vocalizations. These patterns were described as more structured and complex compared to the vocalizations of normal mice.
More structured and complex ultrasonic squeaks.
That's right.
That's quite a subtle but potentially significant difference. So even though we can't hear the nuances, their communication amongst themselves was altered.
Precisely. This suggests that FOXP2 plays a role in refining the way vocalizations are produced, which is thought to be a key step in the evolutionary development of human speech, a move towards more controlled and nuanced vocal output. Beyond vocalizations, they also saw improvements in learning. The mice with the human gene learned new motor tasks more quickly and showed enhanced learning abilities in tasks like navigating mazes.
Oh, wow.
This likely connects back to the enhanced neural plasticity we discussed earlier, making their brains more adept at acquiring new skills.
And what about their physical abilities? The researchers mentioned motor control and coordination. Did the humanized gene have any impact there?
Yes, they exhibited superior motor control and coordination. Think about the incredibly fine-tuned movements our bodies make when we speak, the precise control of the tongue, lips, and vocal cords.
Right.
While mice don't have the same vocal apparatus, this improved motor control in the modified mice could represent a foundational element, a potential early evolutionary step towards the complex motor skills necessary for speech.
Okay, this is all incredibly interesting.
Yeah.
We're seeing changes at the brain level, more complex communication, albeit ultrasonic, better learning, and improved motor skills.
Absolutely.
But let's address the most obvious question.
Yeah.
Did these mice actually start talking? Did they develop anything resembling human speech?
This is a crucial point to clarify. No, these mice did not develop the ability to speak human language. They didn't suddenly start holding conversations or reciting Shakespeare.
Right, because I can definitely imagine some listeners getting excited by the idea of talking mice running around.
Exactly. It's vital to keep in mind that FOXP2 is just one component in an incredibly complex system.
Right.
Human speech and language involve a vast network of genes working together, as well as the development of specific anatomical structures, most importantly, the human vocal tract.
Right.
Mice simply don't possess the biological hardware required for human speech, even with a slightly modified gene.
That makes perfect sense.
Yeah.
So while no talking mice, this experiment still holds some really significant implications for understanding ourselves and our future rights.
Absolutely.
Let's start with how this research might shed light on the evolution of human language.
So when we consider the broader picture of human evolution, this research offers valuable insights into how our unique capacity for speech and language might have emerged.
Okay.
The fact that altering just one gene in mice led to more complex vocalizations suggests that FOXP2 likely played a critical role in early humans developing more sophisticated ways of communicating vocally.
Right.
Comparing these mice with other primates that have different versions of FOXP2 could further illuminate the evolutionary trajectory of our communicative abilities, helping us understand the genetic underpinnings of how we moved from simpler forms of communication to complex spoken language.
So it's like getting a glimpse into one of the genetic building blocks that helped us go from simple calls to complex conversations over millennia.
That's a great way to put it.
What about more practical applications? Could this research have any impact on how we understand or treat speech and language disorders in humans?
This is where the potential for real-world impact becomes apparent.
Yeah.
Since mutations in FOXP2 are known to cause speech difficulties in humans, understanding precisely how the gene functions in these modified mice could lead to a deeper understanding of the biological mechanisms underlying these disorders, such as apraxia of speech. It's conceivable that future research building on these findings could pave the way for the development of new therapeutic strategies, potentially even targeted interventions aimed at addressing FOXP2-related speech problems.
That's a really significant potential benefit. And I noticed the source material also touched on connections to artificial intelligence and robotics.
Oh, yeah.
How does studying a language gene in mice relate to our technological future?
It's fascinating, isn't it?
Yeah.
By gaining a better understanding of how FOXP2 influences learning and communication at a biological level, scientists might be able to develop more sophisticated AI algorithms, particularly for speech recognition and natural language processing.
Uh-huh.
Insights into how genes like FOXP2 contribute to the biological mechanisms of learning sequential patterns could be invaluable in developing robots and AI systems that can interact with humans in a more intuitive and natural way.
Yeah.
It's about understanding the fundamental biological principles that underpin communication and learning, which could then inform the development of more advanced artificial systems.
That's a connection I hadn't immediately made, but it makes a lot of sense that understanding the biological basis could inspire advancements in AI. So to wrap things up, what are the key takeaways from our deep dive into this FOXP2 mouse experiment?
In essence, this experiment beautifully illustrates how the insertion of a single human gene, one crucial for language development, can trigger a cascade of changes in another species. We observed alterations in brain development, specifically increased neuroplasticity, and changes in key regions related to motor control and learning. We saw differences in their ultrasonic vocalizations, suggesting a refinement of vocal output. The mice also demonstrated improved learning abilities and motor coordination. However, it's vital to remember that this did not lead to speech, highlighting the complex multi-gene and multi-system nature of human language.
Right, it's a fascinating piece of a much larger and more intricate puzzle.
Exactly.
This research offers invaluable insights into the complex genetic foundations of human communication and holds potential implications for our understanding of human evolution, for developing more effective treatments for speech disorders, and even for inspiring future advancements in artificial intelligence.
Absolutely.
So a seemingly small genetic change in FOXP2 led to these far-reaching effects in mice.
Right.
It really makes you ponder. Considering this, what other seemingly subtle genetic differences might underlie the unique and fascinating capabilities of different species, including our own, that we are only just beginning to truly comprehend?
That is the question, isn't it?
It's definitely a thought-provoking area. Thanks for taking that deep dive with us.
You're very welcome.
And we'll see you next time.